Good afternoon, ladies and gentlemen. Welcome to the Chulalongkorn University Bridges Nobel Laureate Talk Series. This afternoon, we have special guests for you. Thank you for joining us today. Not only in this conference room, we also going live on Facebook official page of Chulalongkorn University, OIA. My name is Associate Professor Parit Mekrun Kamal. It's my great pleasure to be the Master of Ceremony today. Today, our special guest is none other than Professor Randy W. Schekman. Professor Schekman is a Nobel Laureate from Medicine who received his Nobel Prize in 2013 from Department of Molecular and Cell Biology of the University of California, Berkeley, USA. Professor Schekman has flown all the way from the USA to be with us today and giving us a special talk on the role of public universities on society change. To commence our ceremony today, I would like to invite Professor Jakapan Suthirat, Vice President of Research Affairs, Chulalongkorn University, to give us the opening remarks. Professor Jakapan, could you please give us the honor? Thank you.
Good afternoon. Your Excellencies, Professor Randy W. Schekman, a Nobel Laureate for Medicine in 2013. Okay. Mr. Uwe Morawetz, Chairman of the International Peace Foundation, distinguished colleagues, students, ladies and gentlemen, it gives me great pleasure to warmly welcome all of you to Chulalongkorn University today to attend the prestigious JAPAN-ASEAN BRIDGES event series jointly organized by Chulalongkorn University and the Institute of Peace Foundation. This collaboration dates back to the event series entitled Bridges Dialogue Towards a Culture of Peace, initiated by Mr. Uwe Morawetz as an independent contribution to the United Nations Decade for a Culture of Peace and Nonviolence that took place from 2003 to 2005. Following the resounding success of that series that brought together several Nobel laureates as well as others well-known speakers and artists who all reached out to the vast audience of over 200,000 across the ASEAN, Southeast Asian regions. Bridges has now returned to Thailand and later this year to Japan, with the renewed aim to bringing together the best and brightest minds of the world from November 2023 to March 2024. Nobel laureates in physics, economics, and medicine. The lecture by Professor Randy Schekman today will bring to the conclusion of series that has effectively stimulated academic exchange while enhancing further discussion in several key areas. It's befitting, therefore, that the topic of his talk today is most pertinent to us here, which is the role of public universities as an engine for social change. Today we have gathered befittingly at the Faculty of Medicine and King Chulalongkorn Memorial Hospital to welcome a highly esteemed academics and renowned scholars of global reach, influence, and inspiration. The most distinguished and exemplary Nobel Laureate for Medicine, Professor Randy W. Schekman, whose rigorous research commenced in the 1970s when he studied yeast cells with malfunctions in the transportation system to demonstrate that such malfunctions were due to genetic defects, and this helped expand how different genes regulate different aspects of the transport. These are the efforts that have made him one of the most outstanding celebrities of his generation. He brings with him today's prodigious wealth of knowledge and expertise, as he is best known for the roles he plays in providing a molecular description of the process of membrane assemblies and vesicular traffic of eukaryotic cells. It's due to that basic principles emerging from his studies in yeast are now being applied to study of genetic disease and protein transport. We wish to express our appreciation to Professor Schekman for traveling here to Thailand to share with us his view and wealth of expertise gained from decades of scholarship, research, and extensive academic pursuits. May I also take this opportunity to convey our deep thanks to our extreme business sponsors, BMW Thailand, Dusit Thani Hotels and Resorts, Kasikorn Bank, and Mitsui Fudosan Co. Limited. They have consistently lent their kind support to making this event possible and truly successful. Thank you very much.
Thank you very much, Professor Chakapan. And before we commence on our highly anticipated talk, may I please invite on stage Professor Chakapan Suthirat, Vice President of Research Affairs, Professor Randy Schekman, the Nobel Laureate, Mr. Uwe Morawetz, the Chairman of International Peace Foundations, and Professor Chanchai Sittipan, the Dean of Faculty of Medicine, Chulalongkorn University, for a group photo. For those of you who come from the US, as you can see here in our Asian cultures, we take photography very, very seriously. Thank you very much. And now it's time we've all been waiting for. Mr. Uwe Morawetz, if you can please stay on stage, and may I kindly invite you to give the opening remarks before Professor Schekman's lecture. Thank you very much.
Okay.
So welcome to the JAPAN-ASEAN BRIDGES event series, which is facilitated by the Vienna-based International Peace Foundation. The events are hosted in cooperation with various local partners, including some of the country's major universities, and I would like to say thank you to Chulalongkorn University for hosting our event today. Commemorating the 50th anniversary of official relations between Japan and the ASEAN region, BRIDGES has been continuously held in Thailand and Japan since November last year, involving the participation of Nobel Laureates for physics, chemistry, medicine, and economics. The JAPAN-ASEAN BRIDGES series follows the series of over 800 BRIDGES events, which the International Peace Foundation has facilitated since 2003 to support education in the ASEAN region. BRIDGES has been established as an international, intercultural, and transdisciplinary platform for creative cultures of learning and continued education for all people. The International Peace Foundation has no concept for peace and no fixed solutions how to achieve peace, but we believe that the first step towards peace is dialogue, and the first step towards dialogue is respect. The International Peace Foundation doesn't take sides, but acts as a mediator by creating an independent platform for dialogue where people meet who normally don't meet. People from all walks of life, people who speak different languages, even if they speak the same. The International Peace Foundation believes that the interconnected problems of our world today cannot be solved only by politicians, only by business, only by scientists or by religion alone, but by working together. In the Bridges event series, people from all walks of life meet in a multidisciplinary program to find creative solutions to solve problems and to achieve peace. Peace within ourselves, within our families, within social structures, peace with nature and the environment, peace between nations, cultures, and religions. Peace is a process. Dialogue is a process. It is nothing which can be achieved instantly. It needs time. This is why Bridges is not organized as a single conference, but as a series of events of now over 2 decades in which Nobel laureates have built bridges with leaders in all parts of society and with the general public. Peace is not something which can be left to the elite of a few, but which needs the participation of everyone. Only if many ways cross and people walking these ways meet, can international understanding be achieved and problems commonly solved. If we listen to and learn from each other, we may discover that there is not only one way to achieve peace, but that there are many ways, and certainly ways we have never thought of to go. It is my pleasure to invite you today to listen to and to share your views with Professor Randy Schekman, a Nobel Laureate for Medicine, who has kindly agreed to come to Thailand to help us build bridges. We all look forward to his keynote speech and to his important contribution towards peace. A warm welcome, Professor Schekman.
Thank you very much, Mr. Uwe Morawetz, the chairman of International Peace Foundation. Next, Professor Schekman, whose work we are all familiar with and has revolutionized the field of cell microbiology, and whose vision and philanthropic endeavor has reshaped the field of Parkinson's disease and beyond. Today, he's going to share with us his view on the role of public universities as an engine for social change. Without further ado, Professor Schekman, please give us the honor. Thank you.
Thank you very much. I'm delighted to be here to visit Chulalongkorn University and to begin my brief tour of Thailand. I have had many occasions to visit other countries in Asia, but this is only my second trip to Thailand, and so I'm looking forward to meeting more people over the next few days. I'm going to give you a presentation on my views on the importance of public higher education. I am a product of public education at all levels. In my training as a youth, growing up in California, I had the advantage of a wonderful junior high and high school experience. My high school biology teacher took me under wing, and I was introduced to the opportunity to present my work in local science fair projects. And so I'm going to tell you about the role that played not only in my career, but also in the career of a very prominent Chinese-American scientist who went on to great discoveries of his own. All of these made possible by, in California, the investment, the wise investment by the state in the development of the University of California, which has over many generations provided training for families of middle class and working class children to move on and to advance themselves in many areas, not only science and scholarship, but in the arts and literature, in government and politics, and every other human endeavor. I am personally indebted to the investment that made it possible for me to be here today to tell you about my experiences. So let me begin by telling you a story about another young man who grew up in New Jersey This is the family of Roger Qian. Roger is shown here in the lower right, dressed in a white coat. Roger's family immigrated from mainland China before he was born. He had a family of rich tradition in scholarship. The Chen family spawned many generations of scientists and engineers who went on to great discoveries and achievements in mainland China. His father moved to the U.S. to provide his children with educational opportunities, and Roger developed at a very young age quite dramatic facility in the physical sciences and chemistry and physics, so much so that when he was a teenager, he won the top science fair project in the United States, what was then called the Westinghouse Talent Science Search, #1 in the whole country. He was a budding genius. He went on to Harvard, to graduate school at Cambridge, and he developed tools, fluorescent tools, to allow people studying physiology to monitor the movement of calcium ions into and out of cells. We had the great good fortune to recruit Roger as a beginning faculty member at UC Berkeley when I had been there for some years already. And during his period of time as a colleague at Berkeley, he developed more very essential tools and used these tools with his facility in physics and optics and in chemistry to probe ever more deeply into the active physiology of living cells. He then moved to be an investigator of the Howard Hughes Medical Institute at the University of California, San Diego, where he developed a technique that has truly revolutionized our ability to visualize proteins moving around in living cells. He took advantage of a discovery made by a Japanese scientist in a laboratory in Massachusetts, the Woods Hole Marine Biological Laboratory. This Japanese scientist discovered a protein in jellyfish that had the intrinsic ability to fluoresce. It had no chemical group needed to allow the molecule, once folded, to organize the amino acid tryptophan into a configuration that would allow the molecule, the protein molecule, to fluoresce when exposed to ultraviolet light. Roger, recognizing the value of this tool, was able to obtain from another scientist the gene that coded for this protein, a protein that many of you have heard of. It's called green fluorescent protein. And with his expertise in chemistry and physics, he recognized that the pattern of fluorescence emitted by this molecule might be modulated by the amino acids that reside around the tryptophan residues that would allow the production of various mutant forms of this protein to absorb ultraviolet light and then fluoresce at different wavelengths. And one of the most dramatic images that I've ever seen was the result of a publication of his in the 1980s showing the many different variants of the green fluorescent protein that he was able to produce, each the result of mutations in the interior surface of the green fluorescent molecule that interacted with the electronic configuration of the tryptophan residues so as to produce this spectrum of spectral variants of GFP. Now, the advantage of this and subsequent technology was that it would be possible to use these individual variants in gene fusions to proteins within living cells to visualize their simultaneous movement within the cell to different destinations in the cell. It made possible images such as you see here, where a cell caught in the act of division is labeled with a reagent that recognizes DNA. Thus you see these blue chromosomes, but also to label cells with by adding a GFP variant that recognizes these orange structures that are called microtubules, that are called actin filaments, and then microtubules organized in the division apparatus within the nucleus called the spindle. So these images are produced now and are used routinely in all of cell biology to visualize not dead cells, but living cells as they go through this process of cell division. For this and many other discoveries of his, he was awarded the Nobel Prize in the early 2000s. And we had the benefit of more of his wisdom and genius for several years thereafter, but very tragically he had a stroke and he died prematurely. But he was a genius in so many respects, all as a result, I believe, of the investment of the public in higher education in California. Well, I too was the beneficiary of that, as I indicated at the outset. I grew up in Southern California. I did not come from a family as rich in the tradition that Roger had. My family was, my father was an engineer, my mother was a homemaker. There were no educators, scientists, or physicians in my family. But when I was very young, I had a toy microscope that I used to explore the natural world around. I collected a jar of dirty water from a local river creek and was able to visualize in this toy microscope with just a simple glass slide the microbial world of protozoa swimming and crawling around in my field of vision. It was quite a remarkable revelation for me. And so I saved up my money, and in high school I bought this student professional microscope, a Bosch & Lomb microscope, which became my pride and joy throughout the rest of my high school years. Every year I would mount a science fair project for display at my school and local and state science fairs, and that was how I saw myself emerging from high school intending to be a physician. Here is one such science fair project very early on, my 9th grade science project, all the result of that initial curiosity about the natural world. Well, I went on after this to college and then to graduate school. In graduate school, I learned the craft of biochemistry from one of the great masters of biochemistry of the second half of the 20th century, Arthur Kornberg. From Kornberg, I learned how to take complicated cellular processes apart, piece by piece, molecule by molecule, so that I could investigate the activities associated, in this case, with chromosome replication. After Kornberg's lab, I decided I wanted to do something different, and so I looked into an emerging area of importance in cell biology, and that is the biological membrane. So let me now tell you a bit about the history of what we know about membranes and how membranes are organized and how they function in cells that have a nucleus, so-called eukaryotic cells. This is a cartoon of the bilayer of a membrane as it exists in all cells, from bacteria to human cells. The bilayer consists of two leaflets of molecules called phospholipids. The phospholipids have these red head groups. They are polar hydrophilic molecules. They face either the exterior of the cell or the interior of the cell, the cytoplasm. So they optimize, they maximize interactions with water-filled environments. Interior to the bilayer, covalently attached to the head groups, are two so-called long-chain fatty acids. These are very apolar, hydrophobic molecules. They naturally avoid hydrophilic environments, and when you have a solution of phospholipids just in a suspension with a buffer, these lipids will spontaneously form bilayers so as to optimize the interaction of the head groups with the exterior or interior of a particle, and the acyl chains optimizing hydrophobic interactions in the interior of a bilayer. Now, if it were only phospholipids in a membrane, the membrane would be completely impermeable to almost everything that the cell would offer, and thus it would not have the unique functions that we ascribe to biological membranes. These functions are, for the most part, attributable to protein molecules shown here in these green diagrams. Very often protein molecules extend across the bilayer, where they expose on the outside polar regions to the outside or the inside of the cell. These protein molecules may be channels through which ions or other small molecules pass selectively or unselectively. Or they may be receptors on the surface of a cell that are there to recognize and receive signals in the form of hormones, such as insulin, where this information recognized by the receptor is then transmitted through the bilayer into the interior of the cell to regulate cellular metabolism. One of the most dramatic examples of a cell in our body that has all these features and more is a cell in the pancreas called the beta cell. The beta cell is the seat of biosynthesis of insulin. Insulin is made inside of the cell, and it becomes stored in these little granules, these little eye spots that you see in this image. These are granules that themselves have a membrane bilayer that surrounds an inner core of a crystalline form of insulin. Now, it's been known for many years that all of the protein molecules that are made in cells, those coded by the genes in the nucleus, there are in the human genome about 23,000 such genes, all of these proteins, including insulin, are made inside the cytoplasm of the cell, but some substantial fraction, many thousands of different proteins, have, like insulin, to escape, to be exported outside of the cell. That requires that these proteins somehow be recognized, distinguished from those proteins that will remain in the cytoplasm, and to be encapsulated within membranes, such as is true in the beta cell of the pancreas, and is indeed true of almost all the cells in our body. By the mid-1970s, much of what we knew about how this process is organized was established by a brilliant pioneering cell biologist, then at the Rockefeller University in New York, a Nobel Laureate by the name of George Palade. He was a brilliant scholar, knew many, many things, many languages, but he was a particular expert in the visualization of cells using this instrument, the electron microscope. Now, the electron microscope was designed by engineers in Germany in the 1920s, and it was used to inspect materials that could resist the bombardment of electrons projected down a vacuum central tube, this part of the instrument. The electrons would then be reflected or bounced off the surface of a material, a solid material, and then an image could be collected from this reflection to reveal the surface structure of a mineral or a solid substance. Of course, these electrons were so powerful that they would destroy any animal or plant tissue. It would simply be vaporized. And so what Palade and his colleagues did in the 1950s and 60s was to devise ways of preserving tissues by a chemical fixation, followed by dehydration and embedment of the specimen in a plastic resin. This resin could then be trimmed and sliced to very narrow dimensions using a diamond knife to produce a thin ribbon of sections of the cell, which would then be displayed on a surface onto which electrons would be bombarded. The electrons would then penetrate through these thin slices all the way through clear areas of a cell, but then be bounced off of stained areas of the cells. Particles in the cytoplasm or membranes reflecting electrons would then reveal a kind of a shadow that could be interpreted as you saw in the previous image. These very powerful tools became available for the study of a variety of cells, many of which Palade studied, in particular the cells of the pancreas that are involved in the production of not only of insulin, but of many of the enzymes that are secreted into our stomach to aid in digestion. Palade was able to visualize in the beta cell and in other pancreatic exocrine cells these granules, such as this of insulin. When cells are triggered to export or secrete insulin, these granules migrate from inside the cell to the perimeter of the cell, where the membrane surrounding the granule comes very close and essentially touches the cytoplasmic surface of the plasma membrane surrounding the cell. And at a key moment, then not understood in any detail, these membranes merge by a process that we call membrane fusion, which results in a continuous bilayer surrounding the cell and resulting in the expulsion of this crystalline form of insulin, which is secreted outside the cell. The crystalline form dissolves, insulin courses into our bloodstream, and is distributed within the body to be recognized by receptors on other cells that are responsible for triggering the uptake of glucose that's generated during our digestive process. So these and many other events were now the subjects of Palade and many other investigations. This process of membrane fusion is repeated in virtually all the cells in our body for different roles. Most dramatically at the junction between nerve cells and muscle cells, or in the brain itself. Here, for instance, is a section, in this case of a frog neuromuscular junction, where a nerve terminal is communicating with a muscle cell. That communication is mediated by smaller membranes called synaptic vesicles, vesicles that congregate at the synapse, the junction between a nerve cell and, in this case, a muscle cell. These vesicles carry very high chemical concentrations of a variety of different neurotransmitters, molecules like dopamine or serotonin, or the major chemical mediator of information transfer in the brain, acetylcholine. They carry these chemicals in very high concentration to a point just at the cytoplasmic surface of the nerve terminal where the synaptic vesicles dock, and they are held in register in this position, awaiting a signal, an action potential, propagated down the nerve terminal that causes a flow of calcium ions from the outside of the cell into the inside of the cell, and that explosively triggers the fusion, the membrane fusion of a synaptic vesicle with the presynaptic membrane, resulting in this case in the secretion of chemical neurotransmitter into the region between these two cells called the synaptic cleft. These chemicals then diffuse across the cleft where they interact with receptors, in this case on the surface of a muscle cell, causing eventually the muscle cell to contract. Now this process is going on in the brain, in all of you while you're sitting and listening, in millions of different connections between nerve cells. There are tens of billions of nerve cells in the human brain. Many of these cells make hundreds, and even in one case, 1 million different connections to other nerve cells. So there are literally trillions of such connections in the brain. These connections mediate every aspect of our life, our thought processes, our emotions. Everything about us is dictated by a regulated symphony of interactions culminating or formed by a tightly regulated process of membrane fusion, allowing these chemical neurotransmitters to mediate the flow of information down the numerous neural networks in our brain and in our body. Plotty, inspecting primarily pancreatic excrement cells, was able to deduce, using techniques that now are considered very cumbersome, but at the time were cutting edge, he was able to deduce that molecules like insulin and the other molecules that must be exported from cells are made on platforms synthesized by ribosomes, represented by these green dots, on platforms of membranes called the endoplasmic reticulum, or ER. These molecules, at that point of their production, pass across a membrane, collect in the salmon-colored interior of this structure, and in a series of stages deduced by electron microscopy by the Plotty team, they were able to show that they progress in stages from the ER through a structure called the Golgi apparatus, which had been described first in the 19th century as an unusual structure of unknown function. But we now knew from Plotty's work that this was a kind of a bus station along a traffic avenue. These secretory proteins like insulin are collected into granules, delivered to the cell surface for secretion to the exterior of the cell. I knew Dr. Plotty because he was a close friend of my mentor, Arthur Kornberg. Kornberg had, the year before I started graduate school, done a brief sabbatical in the Plotty lab at Rockefeller University. So they became close. But most importantly, I heard Plotty deliver a lecture in the first months after I began my postdoctoral work at the University of California, San Diego, when Plotty, after having just received the Nobel Prize, flew from Stockholm to San Diego to deliver a lecture at the opening ceremony of the organization called the American Society for Cell Biology. He was a prince in this group of cell biologists. He was worshipped by these people for his technical facility and his brilliance, and so he delivered this stunning lecture describing the pathway that he deduced. But in my case, having just left Arthur Kornberg's lab, where I learned a reductionist biochemical approach to studying cellular processes, I found this image, the image that he showed of this, to be interesting but somewhat unsatisfying. Because in 1974, when Palade won the Nobel Prize, this pathway, though it was clearly of obvious physiologic importance, was not understood in any mechanistic depth. There was not a single protein molecule or lipid whose function could be ascribed to any stage in this pathway. So this here was an enormously complex pathway that probably all cells have, and yet we didn't know anything about how this process was operated. We didn't know about the existence of a channel in the ER membrane through which proteins must pass to initiate this pathway. We didn't know how some proteins were captured into little vesicles that bud from the ER. We knew nothing about how these molecules progress through the Golgi apparatus, how they are then packaged again, delivered to the cell surface, and we knew nothing about the catalysts of membrane fusion. So, at the time, during my postdoctoral period in the years leading up to that, another source for the study of cell biology became of great importance, and that is a simple single-celled organism called Baker's yeast. All of the cell biology that Palade and his generation of scientists did was with animals, or cells from an animal grown in the laboratory. But many geneticists had adopted Baker's yeast as an experimental tool to investigate essential cellular processes. At this time, a great geneticist at the University of Washington, Leland Hartwell, had used genetics to define the genes that a yeast cell uses to progress through the different stages of cell division. It seemed likely to me that yeast would also be a useful experimental tool to map the stages in the process of protein secretion. So let me show you a little bit about what a yeast cell looks like in contrast to a pancreatic beta cell, and the tools that we had available to us to explore the process of protein secretion. So here's a thin slice in the electron microscope of a yeast cell. Not nearly as dramatic as a pancreatic beta cell, but on close inspection, the yeast cell has the same variety of organelles. This structure, seen here darkly staining, is the yeast equivalent of the lysosome. There are membrane tubules characteristic of the ER, largely adjoining the plasma membrane inner surface. In other sections, you can see that the yeast cells have a nucleus. But our attention, my attention, and that of a graduate student whom I'll introduce in a moment, was drawn to this collection of small vesicle-appearing particles that had been seen over the years to congregate under the bud portion of a dividing cell, the very part of the cell that grows during the 90 minutes or so that it takes a yeast cell to grow and divide. Now, it had already been shown that enzymes that yeast cells have to secrete into the environment to harvest macromolecules for their subunits to take into the cell, such as glucose or amino acids. It had been shown that these vesicles carry enzymes that get secreted into a thick cell wall that surrounds the yeast cell. But Peter Novick, one of my first graduate students, and I imagined that these vesicles would be responsible for carrying not only soluble enzymes that get secreted by the cell, but also they would contain the building blocks for the growth of the plasma membrane, such that when a vesicle fuses with the plasma membrane, its internal content would be secreted, but the membrane of the vesicle itself would become added to the growing plasma membrane. A step-by-step membrane fusion process should result in the elaboration of the new surface surrounding the daughter yeast cell. So there is a simple prediction from this hypothesis, and that is mutations that cripple any of the genes required for the production or movement or fusion of these vesicles would themselves be essential genes. If you knock these genes out, the yeast cell couldn't get any bigger, it couldn't enlarge, and it would likely die. So, of course, if a cell dies, you have nothing to study, but fortunately microbiologists over many years had developed simple tools to isolate genes that are otherwise essential for the growth of a cell. The logic is very simple. If you expose a yeast cell or a bacterial cell to a chemical that causes mutations, occasionally that mutation will change a base in a gene to result in the insertion of a different amino acid, an amino acid that may not be compatible with the stability of the folded protein. Such a missense mutation will, not infrequently, introduce a change that causes the protein to be thermally unstable. Now, yeast cells can grow at a range of temperatures, from room temperature up to 40 degrees. But in the range from room temperature to 37 degrees, some of these mutations allow the protein to function at room temperature, but cause the protein to unfold and lose its function at human body temperature, 37 degrees. These are temperature-sensitive conditional mutants, and if the mutation is in an essential gene, then that mutation will cause the cell to die at 37 degrees, allowing, on the other hand, it to be grown at room temperature. So these are temperature-sensitive, conditionally lethal mutations. So very quickly it became possible to collect a bunch of such temperature-sensitive colonies, very simple techniques using Petri plates and toothpicks and simple chemicals, and then to assess among the various mutations, to look for those mutations that specifically cripple the process of protein secretion, so as to distinguish those mutations from others that affect other essential cellular functions, such as chromosome replication or gene expression. So what Peter Novick did was to devise very simple tests to look for such a mutation that causes secretory enzymes that would normally be exported outside of the cell to be retained, to accumulate inside the cell. In the fall of 1978, or in the fall of 1977, Peter had such mutations. He had a mutation that, when the cells were warmed to 37 degrees, caused two different secretory enzymes to build up inside the cell. We studied this, and several months later, as fate would have it, Dr. Pallotti, who then moved to the University of California, San Diego as a professor, came to Berkeley to deliver 2 honorific lectures. And I had the pleasure of introducing him to my new work, but most importantly, the graduate students organized a dinner on behalf of Dr. Pallotti the evening of his first lecture. Peter attended this dinner and described his new results, and Pallotti said, well, now, of course, you must inspect your mutant cells by thin section electron microscopy. We obviously were about to do this, but we raced ahead. And that summer, one of the great moments in my career happened when one morning Peter called me from my office on the top floor of my building down to the basement to excitedly show me an image of the first such mutant called SEC1, after the cells had been incubated at the high temperature for a couple of hours and then processed for electron microscopy. Here is the image of the cell that he showed me. Instead of the very small number of vesicles normally accumulated in the bud of a normal living yeast cell, these cells continue to make these vesicles, but they have nowhere to go. They can't fuse because the gene SEC1 has been crippled, and that results in the failure of these vesicles to fuse with the plasma membrane. Instead, the cell now continues to make these things. They fill up the cytoplasm, and they basically choke the cell to death. So this was a very exciting revelation for us. And over the next year and a half, we collected hundreds of additional mutations, defining initially a couple of dozen different genes whose function is ascribed to individual different stages in a linear pathway. After many years of further analysis, we were able to show a pathway formally very similar to the one that Pallotti had deduced with pancreatic exocrine cells, but with each stage in the pathway populated with a number of genes indicating molecules, protein molecules that function at this step. So SEC1 is shown at the very end of the pathway, but it was, at least initially, one of 10 different genes also required to deliver vesicles for fusion and the final step of secretion. Beyond that, there were many other genes found in my lab and in the labs of subsequent students and fellows. We found genes that encode the channel in the ER membrane through which proteins pass. We found genes required for a bidirectional flow of vesicles back and forth between the ER and the Golgi apparatus. Postdoctoral fellows Tom Stevens and Scott Emmer, in their own laboratories after they left my lab, were able to define a branch point in the pathway that led to the transfer of proteins to this structure called the vacuole. Now, it then became possible in the years after our discovery to clone these genes, to obtain the genes and to sequence them to obtain their nucleotide sequence. Initially, these genes showed no homology to then anything yet in the genome database. But within a few years, the human genome was sequenced, and it became possible to compare the sequence of our genes to the human genome. And not surprisingly, because of the importance of evolutionary conservation, all of these genes were found in the human genome. Excuse me a second. And I use this diagram to show the conservation between the lowly yeast and this important figure, Charles Darwin. Indeed, we now know that 2 billion years of evolution has preserved the essential functions that operate in all the processes that are essential to the growth and division of a yeast cell, are found in the brain and in all of the cells of our body. The SEC1 protein is known to have its equivalent at the synapse and to be required for the organization of synaptic vesicles as they fuse to the presynaptic membrane. Now, knowing this functional conservation, it became possible to exploit yeast as a production platform for the manufacture of clinically important proteins. And I had the good fortune of being able to work with a local biotechnology company growing up in the Bay Area that used yeast technology to introduce the gene for human insulin into yeast. And now, many years later, 1/3 of the world's supply of human recombinant insulin is made in giant fermentation plants, made mainly by a Danish company called Novo Nordisk. And furthermore, yeast is the sole source of the hepatitis vaccine. This same company found that they could take the gene for the surface antigen of the hepatitis B virus, introduce that gene into yeast cells. That gene encodes a membrane protein, and that protein is inserted into a vesicle inside the yeast cell. And for any of you who have been vaccinated against hepatitis B, you've received injections of vesicles prepared from broken yeast cells. Well, that's the adventure that I wanted to share with you today, but it has a happy conclusion. After much work and moving on to studying these processes now in animal cells, I had the call from Stockholm, now almost 11 years ago. Every year, the new laureates are contacted after their initial surprise. They're contacted by the Nobel Museum and asked to contribute an artifact from their discovery that might be on display in the museum. And there was nothing of particular interest from my laboratory that I wished to share with them, but fortunately that old microscope that I had when I was growing up in high school back in Southern California had been saved by my parents. They mailed it up to me near Berkeley when I had a family. Regrettably, neither of my children ever showed any interest in science, and so the microscope sat around to collect dust until I had that call or that email from the Nobel Museum. So I then dusted off the microscope, I shipped it to Stockholm, and if you ever stop by the Nobel Museum in Stockholm, you can see the image of this microscope that was my pride and joy throughout my early introduction to the wonders of life. So that is my journey. I hope there was some source of inspiration for some of you who wish to follow and to make a life for yourself in science. It has been rewarding in many different ways, and for this I credit the the advantage that I had in the education at the University of California. Thank you for your attention.
Thank you very much, Professor Schekman. And now is the time for question and answers. Do we have any questions from the floor? Yes, please.
A lot of taxpayers' investment.
Yes.
And you know, sometimes that gets politicized.
Yes, it does.
And so I think what I got from your talk today is that, you know, collaboration, communication, I guess, you know, clusters of intellectuals pursuing the same goals is probably something very important in a scientifically thriving environment.
Sure. Sir, I'm happy to elaborate on that. In my case, I feel in a way I've been more than simply fortunate. I've been blessed to have some truly wonderful, creative students join my lab over many years. The work that I described was the work of one graduate student, Peter Novick, but I've been very fortunate to have a series of students from all over the world who come to my lab and who shared my enthusiasm for discovery at the basic level. And I should emphasize that all of what I described, though it has had practical application, was driven simply by curiosity. How the cell works, how the cell can do all these complicated transactions and allow itself to be divided and have to repeat this all over. This is the wonder of basic science and the tools that have developed themselves over the years to study these processes at the molecular level. I am convinced that the journey has still only just begun. The wonders of the human brain remain to be understood at a molecular level, and we are now in my lab studying some aspects of that myself, and I've become involved in an international collaborative research effort on Parkinson's disease, a disease that is rising in incidence as the population ages, but for which there are still no cures. And the fundamental mysteries of neurodegenerative disease remain open for exploration at the molecular and cellular level, and there continue to be opportunities to do that for any young person who has the will and the focus to be able to explore the natural world.
Thank you for that inspiring answer and also a very insightful question as well. Any other questions from the floor? Yes, please. The mic is in the back.
According to your cell biology research, in your perspective, what do you think right now is still the gap of this field of the cell biologists?
What are the gaps that remain?
Yes,
yeah.
Remaining gaps.
Yeah.
That you want to write, you want to work, or you want to know still today?
Well, for me, you know, there are always new things, and one of the things that excites me the most that we have been exploring for the past 10 years is the process that cells use, all cells, but particularly our cells, use to export out of the cell intact vesicles. So vesicles move things around in the cell, but it turns out that all cells also produce vesicles that are themselves exported outside of the cell. Our body is filled with billions of extracellular vesicles that are the product of metabolic processes that occur in the cells from which they arose. These are little sentinels that the body sends out and can be used for elaborate diagnostic purposes. So there's a challenge in identifying molecules in these vesicles that can be obtained in a simple liquid biopsy. I think these are going to be tools of great importance in diagnostics of, for instance, metastatic cancer. So we are studying this at a very basic level in my laboratory, and I think there are still many open questions about this. In a larger sense, the brain and the means by which nerve cells communicate along neural networks remains still at a very rudimentary stage, and I think there are enormous opportunities over many decades for talented young people to focus their energies in trying to figure out how to reduce this to the complexity of a molecule or a cell. I think the opportunities are unlimited. It just requires the will to make it happen and the focus to identify an important problem and to just look at it with a laser sharp image.
Thank you.
Yes, please.
Yes, Professor, thank you for your presentation. Would like to know about collaboration among universities. Like you mentioned, Berkeley and San Jose, San Diego.
San Diego, yeah.
Yes, and would like to know the in-depth of the collaboration. Why these make the researchers so successful.
The collaboration among researchers at these universities,
yes.
Yeah. Well, I've learned in my career that it's possible to have one's own laboratory and to explore things as my laboratory did, but as my career advanced and I had more opportunities to interact with other people, I found the additional value of collaboration. For many years I had an amazing collaboration with an Italian scientist who was in Geneva, Switzerland. And after an initial introduction, we spoke virtually every day for 15 years about our mutual interests. He using morphology to study this process, me using biochemistry and genetics. And it was magical. So over 15 years, we published many papers together. We never argued about whose name went where on the paper. It was just because we were both keenly interested in answering the big questions. So I now am so appreciative of that possibility of this kind of collaborative research style that the International Program on Parkinson's Disease that I've had the privilege of organizing is based entirely on collaborations. We have 35 teams in 163 laboratories at 80 different institutions around the world who are in active collaboration on aspects of the basic science of Parkinson's disease, funded very generously by the Sergey Brin Family Foundation in California. So I think that the most difficult problems, the most complicated problems, and neurodegenerative disease is obviously a complicated problem. These require the wisdom and efforts of multiple groups working together in collaborative spirit. So we've been promoting that model.
Thank you very much. And just to touch on that point, I'm sorry, I'm going to revoke my right as an MC to ask a question. From collaborative efforts that you mentioned, for those of us who don't have that luxury of having such a large network, and many of us in the room who are young, enthusiastic researchers, what would be your advice for these newer generations of researchers who are trying to establish themselves and establish those collaborations along the line?
Yeah, so I've thought a lot about what it takes to have a really meaningful collaboration. Academic science has many virtues, but one of them is not promoting collaboration. The reward structure for an academic science favors the individual, his or her little team. It's where your name is placed on a paper and the journal that you publish in. That's all an individual reward system. I benefited from that myself. But for the big problems, you need other people of equal skill and intensity, but who are experts in other techniques. And so for that, there has to be a reward system that favors collaboration. And yet, the usual grant system doesn't necessarily favor collaboration. There are grants that are issued to teams of investigators, but it usually is just a group of individuals who are seeking money and who give the appearance of being a team, but they're still just individual investigators. What we've done in the Parkinson's Network is we built a way of monitoring collaboration and of rewarding specifically collaboration. And so, as we evaluate our teams, we're not simply counting papers in so-called high-profile journals. We're looking at publications that are jointly authored. We're looking at people who are willing to share results with each other within the network before they're published. Not to be secretive about things, they have to share. They have to be willing to share their reagents. They have to be willing to share their protocols. They have to post their work on an archive such as the bioRxiv when it's ready to be submitted for publication. And they have to be willing to publish things in an open access format. So we're looking for people who really have that spirit of collaboration. And fortunately, we found teams that really do imbue that come with that philosophy. And we're working to kind of push that model as best as we can. And we think we're having some success in that direction.
Thank you very much. And as you mentioned a little bit on your endeavor in Parkinson's collaborative efforts, joined with us here today, we are fortunate to have Professor Rungrod, who's a pioneer in our Parkinson's breakthrough clinical efforts in not just in Chulalongkorn University but in Thailand. May I please invite our neurology team to give us a brief comment on such collaborations?
Yeah, how about wait for a microphone?
Thanks very much for the lecture, and I think that we share the same wisdom of trying to combating Parkinson's diseases, in which we know that the prevalence is very high, and we're facing what we actually call it as a Parkinson's pandemic. Yeah. The issues that to approach this problem probably, as you mentioned, really requires a multidisciplinary, collaborative, and importantly has to be long-term and international approach. So I'm just maybe if I could ask you to comment it in terms of at the cellular levels, and we're all aware that before Parkinson's disease patients develop symptoms of tremor, they actually have a very long period of degeneration. We know it's 10 years, 20 years. They start with a prodromal symptoms of constipation, lack of smell, and so forth. What do you think is really happening at the cellular levels? Yeah. And how are we going to be able to probe that? Yeah. And with the hope that one day we're going to prevent or even reverse the pathology of Parkinson's disease. Thank you.
Yeah. I wish I had the chance to have a completely separate lecture on that whole subject, but let me try to distill it for you. To me, the hints that are provided by the genes that we know in familial forms of the disease, there are now over 20 different genes. These genes encode proteins that we know, but we can't put the pieces together in a coherent puzzle just yet. But the genes are important clues. And many of these clues are now being reduced to an understanding of simple pathways in cells that are grown in the laboratory and experimental animals and even in tissues. So let me give you one example that's emerged from one of the teams in this Parkinson network. So one of the genes of the 20 is a gene called LRRK2, LRRK2. It encodes a protein kinase. It's a very low turnover enzyme. It has a very low activity as a kinase. And yet the mutations in families with this form of the disease are genetically dominant mutations that activate the otherwise weak protein kinase. They elevate the kinase activity, maybe twofold, and that is enough to cause this pathology. Now, one of the teams, led by a Scottish scientist by the name of Dario Alessi, has found, in collaboration with one of his team partners, a woman named Suzanne Pfeffer at Stanford, that the activated LRRK2 kinase preferentially phosphorylates a subset of so-called Rab proteins. These are small GTP binding proteins. These GTP binding proteins are responsible for directing vesicles within cells to different destinations in the cell. In fact, the first such Rab protein was discovered in my lab as the SEC4 gene. So a subset of Rab proteins in the human genome, there are 64 different Rab proteins, a subset become phosphorylated by the activated LRRK2. And Suzanne Pfeffer discovered that in cells that are in communication with dopaminergic neurons, these are cholinergic interneurons, those cells have a pathway of communication with dopaminergic neurons called the hedgehog signaling pathway, which is a means by which stressed dopaminergic neurons communicate with these cholinergic interneurons. And the recognition of the hedgehog signal that comes from the dopaminergic neuron is recognized by a structure on the surface of the cholinergic neuron called a primary cilium, a sensory cilium. This has the apparatus that decodes this hedgehog pathway. The phosphorylation of a subset of Rab proteins in those target cells arrests the production of the primary cilium on these cells, which results in a failure of these cells to communicate with one another, which results in the failure of the cholinergic neuron to produce and secrete a growth factor called GDNF that sustains the dopaminergic neuron. So that failure in communication, which can be triggered by simply misphosphorylating a target, results in the death both of the dopaminergic neuron and the cholinergic interneuron. So this is really very reductionist biochemistry, but it bears on the disease because Pfeffer has shown now, not only in experimental animals, but in human subjects who have donated their brain to a brain bank, that these cholinergic interneurons in the biopsies of patients who died of Parkinson's with a LRRK2 mutation have a deficit in primary cilia on their cholinergic interneurons. And at least in the experimental animals, she's shown that you can overcome that deficit of primary cilia by feeding the animal a drug that is now designed to lower the hyperactive protein kinase associated with LRRK2, with mutant LRRK2. So I'm very excited by that as an example. And it turns out, interestingly, that overactive LRRK2 kinase is found in sporadic Parkinson's patients, not only in the genetic forms. So this drug and other drugs that are being developed may well be useful for treating more than just a small number of LRRK2 mutant patients. So anyway, that's one example. There are many other examples of lessons that come from the genes, such as alpha-synuclein, another protein that's implicated in sporadic and some genetic forms of disease. I'm confident that the basic science that's coming out of these works will lead to new drugs. Excuse me.
Thank you. Thank you.
Thank you very much. We have time for one or two more questions. Any other questions from the floor? Yes, please. The gentleman in the middle row.
Thank you for your lecture. It's been a pleasure to attend your lecture. 10 years ago, I have heard that you would start to boycott to publishing a paper to such a luxury paper, such journals such as Nature or other else. It has been 10 years, and does your opinion or your statement still the same? And what to say that you stop sending a paper to a top journal?
Okay. I think what you're asking is another subject that's that I'd love to talk about. It's a longer conversation, but the problem that you're referring to is a general problem around the world, and that is one of the problems I feel is associated with this the reward structure for recognition in science. Typically, the reward structure consists of getting your name in a prominent place on a paper that's published in one of these so-called high-profile journals. And in the life sciences, these are often journals such as Cell, Nature or Science. And I think the problem has gotten much worse over the course of my career. I feel that the commercial influence on scientific literature has exerted a toxic effect on scholarship. These journals are the people who judge the publication of these journals are professional editors who are hired by the publisher to make decisions about what gets reviewed and what gets published. And I feel that these professional editors are in a conflict of interest because they are serving the business plan of the commercial publisher. They are there to make sure that the papers that get published are going to generate citations very rapidly, because the journal judges its success by a number called the journal impact factor that has evolved into a measure of scholarship, a measure that it was never intended to judge. The journal impact factor, which probably most of you have heard about, is a number that was created decades ago to be used by librarians to decide which journals to subscribe for their library. And yet, over now many years, it has evolved into a measure that is supposed to be a measure of success, and these journals sell themselves on this number. I think this has been a terrible influence on scholarship, where we've turned our authority that we had over the decision about what qualifies as great science to professional editors whose judgment we wouldn't seek in any other matter. So when I won the Nobel, I announced on the day of the Nobel ceremony in an editorial published in the Guardian newspaper that I would no longer publish in these journals because of the toxic influence they were exerting on scholarship around the world. And I make this comment wherever I go in the world. It's particularly a bad problem in Asia, where I found that administrators often look at where something is published to judge the quality of the scholarship done by a faculty member or a student, and that is simply a misuse of that information. The name of the journal and the impact factor of the journal should have no bearing on how we evaluate the scholarship. The work has to be judged on its own merits and not on the basis of the popularity of the journal in which the paper is published. So I would urge people in positions of authority to judge otherwise. The impact factor is a phony number. It should never be used by anyone to decide who gets promoted or who gets hired for a job. And I'm happy to deliver that message wherever I go and hope that the message takes hold.
Thank you very much. I have to say I cannot agree with you more, but until I win a Nobel Prize, I probably have to rely on these publishers. Well, we have time for one last question. Yes.
Thank you for your wonderful talk. I think we talk a bit. This question may be beneficial for the audience in the room, as we have people from different backgrounds, some from clinical science and some are basic science. So in your view, what is a part and parcel, the missing piece that prevents translation from preclinical trial to clinical trial? And can you give us the optimism, like the light at the end of the tunnel, as all of us still are very young in our career path? So we just want the guide or the clue that how can we not be like you, but at least better in our career.
Yeah. Well, there's no easy answer to that question. There's a deep valley between basic science discovery and application in the clinic, and that is where private enterprise comes in. I feel that the basic discoveries should be made in the university, where all of that information is available, publicly accessible information shared around the world. But for that message to be translated into the clinic, it has to go through some process of private enterprise where people are willing to invest money in a startup company to explore the application of this knowledge to using targets that come from basic science to develop therapies. And so I feel in the United States we've had a big advantage in doing that because of some wise legislation that was passed in Congress many years ago, when I was a young beginning assistant professor, there was an act published in Congress called the Bayh-Dole Act, which allowed federally funded scientists to patent their own discoveries and then to benefit in the licensing of those discoveries to companies. It wasn't true before that, and so there was no incentive for basic scientists to patent their discoveries. Now, almost all my colleagues patent lots of things, and some of them actually then are taken up by companies, and they turn a profit to the benefit not only of the institution, but also of the basic scientists. So that kind of legislation, I don't know if it's true here in Thailand or elsewhere around the world, but that's been instrumental in the birth of the biotechnology industry in the US. All of that arose because basic scientists saw the opportunity to start their own companies. Most of my young colleagues in my department have started their own companies, and many of them are investing in discoveries that they made in their lab, but now in the private sector. So that's where I think where an investment in basic science by governments and public institutions really has a benefit in every country, not just in big countries. In Thailand, I think the government would be well advised to continue to invest in basic science and to give people like you in this audience the opportunity to benefit from their discoveries by patenting and then taking that technology into the private sector. So that's a path. It's a long path. You know, most discoveries don't lead to something useful. Most discoveries take many years before eventually some small fraction of them generate a drug that reaches the clinic. But that's the gamble that I think needs to be made to advance discovery and to treat diseases that continue to afflict us all.
Thank you.
Thank you very much, Professor Schekman, for your insightful comment. And I believe I can speak for everyone in this room here that we have all been inspired by the fascinating journey and each dots that you have connected along the way, and very informative and comprehensive lectures on cell biology. To end this event, I would like to invite Associate Professor Chanchai Sittipan, the Dean of our Faculty of Medicine, Chulalongkorn University, and also the Director of King Chulalongkorn Memorial Hospital, to deliver a closing remarks. Professor Chanchai, could you please give us the honor?
Your Excellencies, Professor Randy W. Schekman, Nobel Laureate for Medicine in 2013, Mr. Uwe Morawetz, Chairman of the International Peace Foundation, Professor JAPAN, distinguished colleagues, students, ladies and gentlemen. This has been a great honor for us, the Faculty of Medicine, Chulalongkorn University, to host this state-of-the-art lecture by Professor Schekman as a part of the JAPAN-ASEAN BRIDGES event series jointly organized by Chulalongkorn University at the Institute of Peace Foundation. I believe I can speak for everybody in this room that Professor Schekman's elaboration on his own journey toward the discovery of the cell membrane vesicle, trafficking and fusion regulator protein was indeed inspiring to us all. More importantly, on the roles of the public universities and other stakeholders play during this journey, his achievement is a testament that underscores the vital role that we, as educators and researchers, contribute to our society as a whole. The expansion of his ongoing research not only contributes tremendously to the field of cell biology, but it also its critical implication to the potential benefit in Parkinson's disease, which speaks volume to the gravity of his groundbreaking discovery, not only for Thailand, but also an impact in a global scale. In the meanwhile, his visionary influence in stimulating government agencies to invest in basic science is deeply impactful for future generations. On behalf of Faculty of Medicine, Chulalongkorn University, I would like to express our sincere gratitude to our esteemed keynote speaker, Professor Randy Schekman, for visiting us and give us a talk, teaching us and inspiring us today. I would like to also convey a special thanks to Mr. Uwe Morawetz, the Chairman of International Peace Foundation, and Chulalongkorn University International Affairs team, who have successfully organized this event. Lastly, I would like to thank our sponsor, BMW Thailand, Yusitani Hotels and Resorts, Kasikorn Bank, and Mitsui Foundation Group, who has made this exciting educational event possible. Thank you very much.
Professor Chanchai, if you can please stay on the stage and present a token of appreciation to Professor Schekman. May I please invite all of the honored guests to take a one last group photo on the stage. Mr. Uwe, Professor Jakapan, Professor Rungrod, and a neurology team, all associated deans and vice presidents of the universities, assistant dean and assistant presidents of the universities. And this has concluded our activities for today. Please join me in giving a warm, heartfelt round of applause for Professor Randy.